Hi everyone, welcome back to my Scanpy video tutorials. Today I'm going to show you how to find the marker genes and how to visualize your marker genes using important plotting functions that I introduced in my previous video. For today's video tutorial, we are going to use the same single-cell RNA sequencing dataset that we analyzed in previous video, a dataset for human fibrotic lung. We subset and annotated the cell clusters as myofibroblast, fibroblast, smooth muscle cells, and parasites. To identify marker genes for each cell cluster, we use the function in Scanpy package, rank genes groups. In the method argument, you can specify which statistical method that you are going to use for the analysis. You can choose t-test. T-test method is simple and very fast to run. You can use logistic regression method. The online tutorial recommends using Wilcoxon method. You can try all the methods. It will identify very similar marker genes for each cell cluster. For today's tutorial, we are going to run the Wilcoxon analysis. Let's run the Wilcoxon analysis to identify marker genes for each cell cluster. You can see, following the analysis, Scanpy added the marker genes into the unstructured layers as a layer of rank genes groups. It has the gene names, the scores, log-fold changes, p-values, and adjusted p-values. Let's have a look at the data. You can see in the unstructured layers, we have a layer of rank genes groups. If we look at the data inside the rank genes groups, you can see we have the data for the gene names, the scores, p-values, adjusted p-values, and log-fold changes. For each cell cluster, in this dataset we have 4 cell clusters: fibroblast, smooth muscle cells, parasites, and myofibroblast. We can plot the top mark genes for each cell cluster according to their score. Here we show top 25 mark genes for each cell cluster. And you can see we plotted the top 25 mark genes for each cell cluster: fibroblast, smooth muscle cells, parasites, and myofibroblast. And also you can see the mark genes for each cell cluster were identified by comparing the genes in the rest cell clusters. So we can make a data frame, use the data frame function in the pandas package. The data frame will include the information for the gene names, the scores, p-values, adjusted p-value, log-fold changes for each cell cluster. Now we can have a look at the data frame. You can see we have the gene names, the scores, p-value, and adjusted p-value, log-fold changes for each cell cluster. Here is the fibroblast, then smooth muscle cells. And we also have parasite and myofibroblast. Now we can save the data frame as a CSV file named as mark genes. Let's save it and we can open it in Excel. If I open my folder IPF1 nin, you can see we have a CSV file for the mark genes. Let's double click and open it in Excel. In Excel, you can see all the gene names, the score, p-value, adjusted p-value, and the log folder changes for each cell cluster. In my previous video, I introduced some core plotting functions. We can use those plotting functions to visualize top mark genes for each cell cluster. For example, we can use the dot plot function to see the expression level of top 8 mark genes for each cell cluster. We can also use the dot plot to see mark genes for each cell cluster that have at least the 3-fold higher expression compared with the rest of the cell clusters. I introduced the wireline plot last time. We can use the wireline plot to see the expression level of individual genes. Here we use collagen 1A1, collagen 3A1, and CTHR-C1 as examples to show mark genes for the myofibroblast cluster. You can see collagen 1A1, collagen 3A1, and CTHR-C1 are highly expressed in the myofibroblast cluster. And in my previous video, I also introduced the stack wireline plot function. And we can use the stack wireline plot to plot lots of genes in the same figure. You can see here we have 8 genes for each cell cluster, and we use the stacked wireline plot to plot all the genes in the same figure. In my previous video, we didn't use the split plot wireline function. Here, with the information of rank genes groups, we can compare mark genes using the split wireline plot function. If we run this function, you can see the expression of mark genes for each cell cluster were shown on the left-hand side in blue color. On the right-hand side, the brown color shows the expression level of those mark genes in the rest cell clusters. So we have 4 cell clusters. The first figure shows the top 20 mark genes for the fibroblast cluster against the rest cells. The second figure is for the smooth muscle cells, and the third figure is for the parasite cluster, and the last one is for the myofibroblast cluster. Heat map is another option to show the expression of those mark genes in different cell clusters. Let's plot the top 5 genes for each cell cluster in the heat map plot. You can see the high level of expression for the mark genes in red color for smooth muscle cells, parasite, fibroblast, and myofibroblast. If you want, you can also run the tracks plot. Here, the gene expression level was shown by the height, and also we introduced the matrix plot. Let's run the matrix plot for the top 8 genes for each cell cluster. Because the myofibroblasts are the pathological cells in the fibrotic lung, we can also focus on the comparison of mark genes between the myofibroblast and the fibroblast. Here we use the dot plot as an example. You can also try other plot functions to plot the marked genes for your dataset. So we knew that the myofibroblasts were derived from resident non-fibroblast. We can run the rank genes group function to identify differentially expressed genes in the myofibroblast cell cluster against the fibroblast cluster. Let's run the rank genes group function and plot the top 25 genes for the myofibroblast cell cluster. So you can see we plotted the top 25 genes for the myofibroblast against the fibroblast cluster. You can see CTHR1 genes for the collagen isoforms and the ACTA2 are the genes that are highly expressed in the myofibroblast cluster. If we run the split wiring plotter now, you will see it only shows the myofibroblast cluster against the fibroblast cluster. Okay, that's it for today's video tutorial. I showed you how to identify marked genes in Scanpy, and also we used some core plotting functions to visualize the marked genes. I hope today's video tutorial could help your data analysis. Please subscribe my channel if you like my video tutorial. Thank you, and I hope to see you in my next video.